Hello, Mateusz. Can you hear me? Oh, yes.
Yes, I can hear you.
It's okay. Do you want to check? So I'm Clara Altin-Toprak from Bioregio Stern. Do you want to check if your presentation works, or is everything clear for you?
We have checked yesterday if the connection will be okay, so I'm actually ready. There is no need to further checking, I think.
Okay, then it's fine. Okay, then we are waiting until 11 and then we will start.
Okay.
Okay.
Ouais, ce qui est génial, en fait. Moi, je n'avais jamais réalisé ça avant peu de temps. Et quoi Oui, bah oui, sans doute que c'est un peu vrai, mais c'est pas grave, quoi.
Allez, pour quelle
So hello, I think we should start with the webinar so that we are on time. And I would like to welcome first to the webinar on 3D printing of medical devices. My name is Clara Altin-Toprak. I'm from Bioregio Stern in Germany. And it's a really great pleasure that there are so many interested in additive manufacturing. And we are really happy that we could win 3 experts of the 3D printing area. They will give a broad overview on 3D printing and how it can be applied in medical technology. Before we start, I just want to outline some rules. So please notice that the camera and microphones are switched off except for the speakers. And you can ask questions during the webinar via the chat function. And if you cannot hear the speaker, please inform us via the chat function or use the dial-in number. Here's one example for the for France. And now we come to our agenda. So first, Emily Romeo from Lyon Biopol will give an introduction of the S3martMed project. Then Kirsten Borchers will give an overview of bio-based materials and how modified gelatins can be used for printing, spraying, and coating. Next, Christophe Marquette will present the French 3D Fab, which is located in Lyon. And the last talk is about 3D printing and rapid prototyping in industry and is given by Mateusz Pawlik from Carbiomede in Gliwice. So at the end of this free presentation, we will read out some of the questions which are asked via the chat function, and they will be answered by the experts. And so enjoy this webinar, and I hand over the microphone to Emily. So please,
Okay.
Emily, could you? Yes, okay.
Thank you very much. So I'm just changing the screen, share my screen with you. Okay, so thank you very much, and good morning, everybody, and welcome to this webinar. So I'm Emilie Romeo, I'm the coordinator of the SmartMed project, and I will give you a very short overview of this initiative. So SmartMed is a European initiative that is supported by the European Union in the COSME program. It has started in 2018 for 2 years. And so the initiative gathers 5 cluster organizations around Europe dedicated to healthcare and medical technologies. So as you can see, these organizations are Lyon Biopol in Auvergne-Rhône-Alpes, WEBINAIRE in Piedmont, BioWin in Wallonia, and BioRegiostern in Bad Württemberg, and Med Silesia in Upper Silesia, Poland. So our ambition altogether is to build a strong partnership in order to boost scientific and business collaboration between SMEs and the research technological organization in medical technology across our different regions, and also to encourage the most promising collaboration ideas to become a real project, thanks to the support of the different cluster partners, and also in collaboration with the regional authority and the European Union. So in order for you to identify synergies and generate this collaboration opportunity, during the SmartMed project, we plan to organize webinars. So in order to promote the different competence in our region. So the next webinar will be on July the 4th on artificial intelligence in medical device. And also at the moment we are preparing our first interregional networking event, which is named SmartMed Needs Smart Tech. So it will be held in September at the University Hospital in Tübingen. And during this event, you will have the possibility to see live surgery session where clinicians will present their challenge and needs in medical technology, so in real condition. And also the participants will have the possibility to arrange face-to-face meeting and have a brainstorming discussion. So the registration will be open in June. So we will give you, of course, the information. And if you have any interest, please contact us. And again, yeah, the purpose of also of our activity is to support the different players that would like to build project to go a step further, supporting you to find the right funding scheme. So if you have any question, here you can see the different contacts in the cluster organization. So please don't hesitate. We also have a website where you can find all the information. And so now I will give you the floor to Dr. Kirsten Boscher, and I hope you enjoy the webinar.
Thank you very much for your introduction. My name is Kirsten Borchers, and I'm working in Stuttgart at the University of Stuttgart, and I'm looking for my screen, sorry. Okay, now I want to give you an introduction into current projects concerning bioprinting and our contributions to the development of bio-based biomaterials, in particular modified gelatins for printing, spraying, and coating. I would like to start with a short introduction into 3D printing in general, which is also called, or better called, additive manufacturing, because not all technologies are really related to a printing process. The workflow of additive manufacturing is always that you start with a 3D data set, which can be acquired either from a CAD software program, can also be a surface scan, which is already used, for example, in dentistry, or it can be 3D data from medical imaging technologies like tomography. These data can then be manipulated on the computer, and the pictures are then transferred into a format that can be translated to a technology that then will build the physical object from these data in a layer by layer production process. And now these layer by layer additive process can be of different kinds. It does not have to be a printing process, as I said. It can also be that a liquid resin, a cross-linkable liquid, is layer by layer cross-linked, solidified. It can be that liquid is printed layer by layer and then cured or solidified by a laser process. It can also be that a polymer paste is extruded layer by layer, or last but not least, other technologies fuse powders either of polymers or metals, also in a layer by layer process. So if you look at this examples, you may get an impression from the workflow, starting from the digital model and then going to the physical object. It is important to recognize that the so-called 3D printing process is always a surface technology. So the data that is transferred to the physical object is always the contour of the surface, and then just filled by material, which is here seen as a green filling. So you can not have the data of, as in this example, a skull with the brain inside, and then start the printer and print bone and brain. You always have the information on the surface, and the printer will form this surface. And if you have an inner and an outer surface, the printer can also fill the space, but only by a material, a filling material, not by the real structure that is inside, for example, the skull. So this technology has been used for quite a time, for example, to produce models in medical technology, to plan surgery, or to pre-adjust prostheses to a personalized model, as you can see in the middle, or to help surgeons with special guides to perform very complicated surgeries. In these cases, of course, the material which is used to form or to produce the metals is not very critical in terms of biocompatibility or other requirements. If you think one step further, which would be producing not models but functional products, as for example orthosis or prosthesis, or even orthopedic implants, of course the requirements to the materials that are used are much higher. They have to fulfill the medical standards for implants, and at the same time they have to be suited for processing by these additive technologies. So this is a much higher challenge than producing models. when we go in our visions now, again, one step further, the idea is if we can use these additive manufacturing techniques to produce one day even biological implants by using biological materials and cells, and try to reproduce personalized tissue implants based on the medical diagnostics, as for example tomographic data. There are various techniques which are suited to process biological or bio-based materials, and of course there are such technologies which are low temperature processes and technologies that can process aqueous solutions, so water-based solutions, which are the standard material if you work with biological materials or even cells. This can, for example, be laser-based techniques such as the stereolithography, where you have a liquid that can be cross-linked by a laser pulse, and usually these techniques are able to produce rather high resolved objects. If you think of real printing technologies such as inkjet printing, you can also produce rather high resolved objects, but based on the fact that the droplets will always flow when they meet the substrate, it is very hard to really produce precise structures. Another technique that is usually applied for bioprinting is the extrusion or dispensing, or also called bioplotting technique, where a hydrogel or a paste is extruded from a nozzle, and as these fine filaments are not very, very tiny, the resolution is not so high that you can achieve. Now, facing the different technologies that can be used for bioprinting, we need to adjust the materials that should be processed to these processes on the one hand side. So we have the level of the solution that has to be adjusted to the process. And on the other hand, after solidification of the material, you have the hydrogel, which should be adjusted or optimized for the cell type, the type of tissue that you want to reproduce. So you always have these two levels that have to be addressed by if you want to formulate a bio-ink, a material that can be processed by additive manufacturing on a biological base. We use, or a material which is used by many and evaluated currently by many groups, is gelatin, as it is a material that is derived from native tissue, from the collagen of tissue, but other than the collagen itself, it is water soluble at physiological pH, and as that it can be used in a solution for printing. But as you probably know from gelatin as a cooking ingredient, the drawback of gelatin is that it forms hydrogels at low temperatures, but it melts at higher temperatures. And as such, it is a solution also at body temperature. And if you want it to be stable, you have to perform a chemical cross-linking additionally to the gel formation. If you want to biofunctionalize these gelatin hydrogels with or in view of different tissues, you have to add additional biological components such as hyaluronic acid or chondroitin sulfate, or also minerals for bone inks, and as that you need additional cross-linkable biomolecules also. So what we do is that we modify these biomolecules with functions that can be cross-linked on the one hand side, and additionally with functions that just mask certain functions of the gelatin molecule without being chemically reactive. And by this we can address, as I will show on the next slide, we can address the properties of the hydrogels and the properties of the solutions separately from each other. And by adjusting the ratio between cross-linkable and masking functions, we achieve a whole portfolio of different gelatin derivatives with different properties. We can control this modification process very nicely, and as you can see here, for example, we can produce derivatives with low or medium or high degrees of modification, and we can then decide whether we have only one kind of modifying functions within the gelatin or different functions like the cross-linking and masking functions in different ratios. We have also developed a method to determine the absolute or the total amount of functions we introduce into the gelatin. So we can say in millimole methacrylic functions or millimole acetic functions per gram gelatin, how intensive the degree of modification is of the diverse derivatives of biomolecules. By doing so, we can control the properties of the polymers on the one hand side, like the isoelectric points of gelatin, the electric charge of the molecule, or the hydrodynamic radius, which is something like the size of the molecules. And this again will affect the intermolecular interactions and then also the solution properties. So the hydrodynamic radius, for example, will affect the viscosity of solutions or the gelation temperature. And last but not least, we also, of course, can then adjust the hydrogel properties by the amount of cross-linkable functions that we use in terms of the stiffness of the gel, the swellability of the gel, or by changing the composition, also biological functions, as I said, by addition of other biological components. So now I would like to give you some examples of what we of projects which we did with the different bioinks, with formulation of different bioinks, and in particular I will show you an example for a bone-specific ink, a vascularization ink, cartilage-inspired matrix gradients. Then I will show you that we can also achieve low viscous solutions that are even suited for inkjet printing. We used our materials also for pro-angiogenic coatings, and we can show that spray drying becomes easier or possible with higher concentrations when we use modified gelatins. The first example is an ink that was formulated with view to reproduction of bone tissue, and this means that we have a gelatin-based hydrogel ink, which also contains a mineral component, the hydroxyapatite powder, to stabilize the function of the cells, which we added which are stem cells that can, in these inks, differentiate into bone cells, the osteoblasts, and as these cells are in the BioInk 3D environment, which is adjusted to their needs, to their specific needs, the cells start to produce the bone-specific matrix by themselves. And this is always the idea of these bioprinting approaches, that you start with a material that supports the cells that you use to produce the tissue-specific matrix, to reproduce the tissue by themselves. So we do not print the ready-made tissue, but we print cells that should then be able to reproduce the tissue by themselves. In this case, we formulated the ink such that we can use an extrusion-based technology, and we adjusted the gelation temperature of the ink and the viscosity such that we achieve these self-supporting filaments, so that we can print layer over layer without the material melting on the substrate. Another example is an ink that was optimized as to form a very soft hydrogel and still be stable enough to form a 3D object, in order to achieve that cells that form blood vessels, small capillary network fragments, to move and to migrate within the hydrogel, so that they could form these vessel-like or capillary-like structures. So we start with single cells which are distributed within the ink, and if we have these hydrogels, which are these pink hydrogels on the right, which are very soft, as I said, these cells start to migrate and follow their genetic program to form these precursor vessel-like structures. This is not possible if the hydrogel is too stiff, too strongly cross-linked. And here you can see a combination of the bone ink and the vascularization ink. And this is in the view, or in view, to provide a precursor tissue that can then be remodeled to a vascularized bone tissue in a bioreactor, for example. By using different kinds of bioinks with different composition, we can also produce gradients, of course, by layer application of these inks. And in this case, we used different concentrations of hyaluronic acid and chondroitin sulfate with the gelatin-based inks, and to reproduce a material gradient as it is in the native cartilage. And we can also, as you can see here, add cells to the ink, and after printing and incubation, we see at the green color shows the live cells and the red color dead cells. So the cells always are vital after printing in the 3D inks. We can, with these additive techniques, of course, form individualized morphologies, anatomies. This is only an example. Nobody needs a hydrogel nose, of course, but it is a quite complex structure, and you can see here, for printing a structure with such cavities, you need a special or an additional supporting ink that will then, after the printing process be removed. And this is an additional challenge to have these kinds of materials that can be combined with the bio ink and then be removed after printing. We can also formulate inks that are not viscous, like these, what you saw just on the previous slides, but which are very low viscous, so that such that they can use in a drop-on-demand process like inkjet printing. And by this, we can, for example, produce these little pads of hydrogels, which are the small dots here on the hydrogel substrate. And within these dots, we can also cultivate cells. Yes. Then the another application for the gelatin-based materials, which is not really for an additive technique, is that we can use them for coatings. And here you see an example where we have a 10 micron coating, the dark blue, the dark blue coating on this thin PET membrane. And these coatings have been used as an storage for growth factors, in this case, the vascular endothelial growth factor. And you can see in this graph that from these thin layers, we can measure the release profiles of the growth factor. And if we test the coatings in an CAM assay, the assay of in a chick egg, you can see that without the growth factor, we have less development of vascularization within the egg than if we have here the coated membrane with this growth factor. Again, here for another coating material, the amount of small capillaries increases if we incubate the growth factor loaded coating on the egg yolk. Last but not least, we have the opportunity or the possibility to spray dry gelatin solutions. And just to give you an idea, if you use native gelatin solutions, which have the which tend to gel, and therefore you can only use very low concentrations of gelatin in this spray drying process. And if you use modified gelatins with reduced gelation, you can really increase the mass concentration in the process, and you will be able to produce small particles, load them with active ingredients, etc. So to sum up, with the biomolecules from the native extracellular matrix, we can formulate bioinks or biofunctional solutions by modification, either with cross-linkable groups, which are used to form stable hydrogels in aqueous environments at body temperature, or modification with masking groups, which reduce the gelation and gelation of gelatin, for example. We have a method to characterize these derivatives in terms of the total degree of modification. We can control solution properties and hydrogel properties, and we can formulate solutions or inks either for bioprinting, tissue-specific bioinks, or solutions and or inks which are adjusted to specific process requirements. With this, I would like to end my presentation. Thank you for listening, and you can ask questions, of course, later on. Thank you very much.
So thank you very much, Kirsten, for this very impressive talk. And now we go to the next speaker, Christoph from the 3D Fab platform will present this platform to you.
So, just starting the presentation. Good morning, everybody. I'm just waiting for the full screen. Yes. So I think you get all the full screen now. So, yeah, good morning. So I'm gonna make a short presentation of a technological platform. So this platform is a consortium here in Lyon between 3 different laboratories. These laboratories are either focused on tough material like ceramics, either on biochemistry and chemistry, or on pure photochemistry. Okay, so and we get all together in a particular platform that have the facilities to print and bioprint. So I will show you the structuration and the facilities we have. Trying to go next slide. It's slow, very slow. Oh, yes. We have been founded in 2015, and we have now 2 locations, both in Lyon, but not at the same place. So we are located in 2 Axel 1 buildings, and one is campus, the top one, which is where we are making the collaborative innovation and collaborative research. So it's basically research between private companies and academics, or academics and academics, and the scale up, the bottom one, which is a scale up building where we can manage to make some industrialization of 2D printing and scale up of the processes. And in this second one, you don't need to be in collaboration with a company. We can only be a company. So the platform is, as I said, was founded in 2015. So it's based on the funding from the region. So it's local, but it's funds from the European state, okay, and from FEDER. And we have something like 1 million annual budget. Half of this budget is from governmental funds. The other half is almost equally distributed between private contracts and collaborative contracts. Collaborative contracts mean with labs in France and in Europe. Okay, so now the tendency is that more private contracts are coming in, and we get less collaborative with academics, but still we have some strong connection with the academics. The main information is that this 1.1 million budget is almost fully dedicated to buying and building and prototyping some machines for additive manufacturing and 3D bioprinting or 3D printing. So we have the capacity to invest for each project, to invest half of the money that is needed to prepare or to change or to modify or produce a new machine. Okay. We have a strong network. So we are academic-based platform, but the network we have, because of the funding we have, the network is with private companies mainly. So there's different level of interaction. Some are pure users, some are partners. We partner are the one that create the platform with us. So some of them have their machine here, some of them were at the beginning, and we have also contacts. So you can see that the big picture is that there is a very different field of application. the main core is healthcare and health application, but you also have some application in, for example, luxury cosmetics, some application also on pure chemistry. But the center, the core is healthcare application for the platform. And we also have locally a network of laboratory outside of the platform with whom we are working on development of additive manufacturing techniques or formulations or concepts. So some are pure chemists with labs that are making some pure organic chemistry or catalyzer or things like that that are useful for additive manufacturing. Some are pure material and polymer designer or researcher, mainly on the ceramic and the polymer characterization. So all the mechanical properties and so on are done in these labs. And since last December, we have a very big area in the clinical applications because we have signed a special agreement with the hospitals plural in Lyon, with whom we have an agreement on the capacity of the platform to produce some medical device for clinical trial directly here at the platform. So that means that we are validated as a production line for medical device because part of the platform is inside the hospitals. So this platform can be used for clinical trial and clinical application. And this is a very strong point because then we have a direct transfer of what we will do, for example, in material like ceramic. Through the platform, we can go directly to clinics if the ethical committee and the regulatory affairs are cleared. But we have this complete pipeline between material development, applications, and then clinical application and trial. So that the platform is focused on projects located between TRL 1 and TRL 6. Why I show you this part is it's because so obviously we are academics, so we are going down to a TRL 1 from the proof of concept, but we are not going higher than TRL 6 because the meaning of the platform is to make some innovation and maturation of projects with either with the private companies or the labs, but no production. So the only production we will do is the production for a clinical trial, not for commercialization. So then, after TRL 6, usually we have patents or prototype of machines or new processes, and here we are transferring technology to the private company that was the partner at the beginning. So we are in this area, and during this project usually we have some formulation activities, prototyping activities of the machines, and validation and so on. So we have 5 main offers in the platform. So we have projects on bioprinting for sure, some project on 4D printing. I will not talk so much about that today, but some projects in 4D printing are focused on the healthcare application. We have a pure optical offer, ultra high resolution and holographic printing, which are very technological and very applied to more than the healthcare. For example, ultra high resolution we apply a lot on surface texturation, optical component production, and so on. The holographic printing, it's a part dealing with a new technology called development of the platform itself. So it's basically based on patents we have. And the ceramic printing, it's a part about bone printing using tough material. So the activities are then segmented in field of application. So the first one is the biomaterial for sure. So here we have a lot of project on companies coming with new or already validated biomaterials, and they want to know how to print it. Is it printable? Can we print it with a new process? Is there a new process to invent to print their material? Then we have a strong focus on polymers because we are making a lot of reformulation and process development for polymers, either implantable polymer like silicone or polymers, very technical polymers for other applications than the healthcare. Then we have Lux activity, which is highly confidential, but which is also very interesting in terms of material that need to be printed, and also in terms of regulatory and environment around the material and the final product. And then we have this clinic interface, which is very strong and which require the ISO 13485, in which we are involved now. So the platform shall be validated and certificate at the end of this year, in order to be able to produce this medical device for clinical application and clinical trial. So just I had this slide just to show you what is needed in this kind of platform in terms of expertise, because you've seen we are working on the formulation, the processes, the biology, the regulation and the quality control and so on. So you see that the people here, they are from different areas and different expertise, and this is really something we need to gather in order to be able to bring all the expertise in one site, in one particular platform. So just there's a few slides about the different infrastructure or technologies we have. So this is one of the first printer we designed with a private company which is called LabSkin. So now this printer is used for skin bioprinting for cosmetology testing instead of testing on animals. So this is an extrusion based system, and it's using gelatin plus other component, bio ink for production of full skin bioprinting construct. So this is similar machine, but with more R&D focus. So we have a lot of different options inside. This is open source. So all these machines are open source. We are modifying them ourselves together with a French company. So every time we need to add a new component, a new add, a new function, then it's easy for us to change something. So this is the R&D bioprinter we have, with a lot of different temperature control, sterilization, and so on. This one is one we bought from a US company, and we developed the bioprinting part and the in vivo bioprinting part. I will talk about that later in the platform. So this is a robotic arm on which we have grafted our particular bioprinting head, in order to be able to print on non-flat surfaces, because when you go to very complex shape and very complex objects, then you will need to print something on something that is not flat. So this is not a Cartesian printer, it's a 6-axis printer. This is a specific machine for ceramic paste. So it's a ceramic printing using very highly charged polymer, so a lot of charge of ceramic charge inside. So this is particularly applied to the ceramic because the motor and the extrusion power is very strong, and you can print some very thick pastes. Another ceramic printing based on stereolithography. So here you have also very thick paste of photopolymers charged with a lot of mineral particles, and this is very strong for us because this machine is validated for production of ceramic implant. So we have the capacity now that we have this machine to produce some ceramic that can be directly implanted in clinical trial. So this is why we choose this one. So using this machine, first you print using stereolithography, so photochemistry, and then you sinter everything in high temperature, higher than 1000 degrees. So you burn every organic component and every trace of the photo initiator, and then you get your ceramic. Then you have plastic prototyping. I will go fast here. So this is just for us to modify the other machine. So we are making the prototype using this kind of printer for the plastic parts. We have a very large printing setup also that can print 1.6 meter cube meter. So here it's a sculpture, but this is not the main area of application. This machine is used for all the prototype or all the plastic parts that are too big for the classic machine. So we are making a lot of prototype for healthcare, like specific prostheses or things like that have large shape and large size. We are using this machine for do that. And then we have also parts machine for light-based printing or additive manufacturing, so DLP and ultra high resolution. This ultra high resolution is from a French company near Lyon called Microlite, and this one is very interesting for us because the resolution is below the size of a cell. I think I have a few slides about that later on. So I will now show you a few examples of what type of project we are hosting here at the platform. So these projects are either project developed by the platform itself or project developed in collaboration with other labs. So for example, this first project, which is bioprinting in surgery theater. So the idea is to be able to bring this robotic arm in the surgery theater, or at least the same concept, and to print directly skin on skin injuries, particularly here severe burn patients. So this is a development with a company and another lab. So you can see their name here, LabSkin and Imopa. So the basic concept is that topographic scanner is used for getting the image of the injury here in blue, and then in the surgery time, so not getting out of the surgery theater, directly in the surgery time, a biopsy is taken and blend and disperse into a bio ink in order to make an autologous bio ink, and then this bio ink is loaded, just like you see in the movie here, is loaded into the cartridge of the bioprinter and print directly using the topography we have from the previous images. So this setup is supposed to stay in the surgery theater and is supposed to be very fast. So the skin is fast. Skin and autologous bio ink preparation is 15 minutes. All right, and then the printing, depending of the size of the injury, will vary, but we said that for 20 centimeter per 20 centimeter, it's taking like 20-25 minutes. So half an hour, 1 hour, you will be able to print back these cells dispersed in the bio ink. So how does it work? the idea behind this is that we were able before to print some skin in vitro to recapitulate skin structuration, the different layers, dermis, epidermis. So now we are also able to make the hypodermis. So we were able to do that with two different inks. But so here you see the difference between human skin, and so the human skin mean biopsy, and the bioprinted skin. So obviously there is some difference, but obviously also you have some similarities in organization and composition. Also the composition here for the skin, bioprinted and human. So well, I will not go in details. You can go see the publication. Well, all the extracellular matrix you want to get are here, but this is in vitro, not in vivo. So this approach was printing the different layers on top of each other. The in vivo approach is blending the full biopsy, printing it on the injury, and leave this reorganize itself on top of the living body. So that's one ongoing project. The next project is based on silicone. So here, one more time, we have some patents here. Some patents are about the formulation, other patents are about processes to process silicone with different viscosities and very low viscosities and so on. So we are working a lot in this area with Elchem silicone. So you can see here, and we are in the consortium here where we are focused on building some prostheses for the airways for people after a resection of this trachea following either accidental or cancerous resection. So we are printing very complex objects. So you see the video here. This means that you need to work a lot on the formulation and rheology and also processes to be able to print this very complex and highly texturized inside the implant in order to be able to produce them. So the idea here is that we need to produce this implant. So this is just this part inside this implant according to the patient anatomy, to the patient surgery, former surgery, and so on. So this is applied to personalized medicine. Okay.
So excuse me for interrupting you, but you are a little bit over time.
Sure.
Maybe you can. Yes.
I have
okay.
3 slides.
Okay, that's perfect.
So this is projects about bone printing. So obviously the idea is to segment from a scanner images and then generate a particular implant and re-implant this. So we are working on the maxillofacial because it's a very challenging bone, very highly constrained bone. And finally, we have some projects on surgical models. So here one project about tumor resection in pediatric surgery. The same here, pediatric surgery. So we applied what we know about silicone printing here to make the lung inside this model, a very looking with the mechanical very similar to the line that you can have in vivo. Okay, and this is what you can have with the very high resolution. So you see 10 micron part here, so the size of a cell. So basically you can have these kind of things with a cell inside if you want to capture some cells or deliver some cells somewhere. So I will go fast. So thank you for your attention, and if you have some questions, so here are my contacts. You can call me or send me an email. Thank you very much.
So thank you very much, Christoph, for this really broad and fascinating talk and all the competences from the 3D Fab. And now we go on with the view of the industry from Mateusz. Please, could you tell us something about the 3D printing from your company?
Yes. Hi, can you hear me properly?
Yes, perfectly.
Okay. I'm Mateusz Pawlik from Cabiomed, computer-aided biomedical engineering. So, point of view from the industry, I would say, and our project, some case studies, not only related to 3D printing and rapid prototyping, but this a bit longer title. So, R&D on demand for medical, sport, and rehabilitation industry by means of 3D printing applications in a way of custom-made mass-scale manufacturing. So quite a contrary as itself, because from the one hand, custom-made, from the other hand, mass-scale manufacturing. But I think you will understand what I mean exactly, or you will get inspired somehow by our projects. And maybe it will give us at the end some nice cooperation. So what is Cabiomed? We are R&D on demand department to hire for medical sport rehabilitation industry. I have a team with background and experience in medical engineering, mechanics, material engineering, manufacturing, and continuous improvement. We are cooperating with local technology and medical universities. We are completely commercial company working with universities and some public institutes, but our projects, our R&D activity is fully commercial. What does it mean, R&D on demand, from our point of view? We are providing services of R&D outsourcing, one-time projects, and we have some of our own projects related to medical devices industry. We have activities, typical engineering activities like computer-aided design, finite element analysis, mechanics, optimization, but also this not so nice side of the business, so documentation according to ISO standards. Full quality documentation, providing quality systems, launching for small startups, etc. And going to our projects, for example, we are conducting early prototype of a tissue adhesive dispenser used by one of cardiosurgeon from a local hospital. The idea was to create at least idea of the device which will allow to use tissue glue, which is really dense, which has really high density, and the strength needed to use it is really high. Whereas it was needed because of the surgical procedures related to vessels, and now it's in an early prototype phase, but we have found some investor for this project to go from prototype to functioning prototype and tests. So as for now, the idea was okay, and now we are going to test it in a clinical trials. We start device. So the device which main aim was to allow people with disabilities to leave their homes because of those last 3-4 steps, which are impossible to go through without such a device. The idea of our customers, because in those projects we were a subcontractor, and the idea was to create the device which will be mobile, lightweight, with quite long range, possible to be driven on a battery, and which could be attached to almost every wheelchair. We have started with full documentation, because in our European Union it's a class first device, whereas according to FDA it's class third. So it was really needed to start with quality assurance and quality documentation to provide the device which will fit into further standards. Because if we will design it and it will fit only in our local Polish market, the idea was not here. It was an idea to provide a device which will be sold worldwide. Our models are mostly focused on a veterinary market as for now. Our own services, our brand is mostly dedicated to veterinary industry in Poland and in Central Europe. as we have found that there is a huge need of surgical helps, surgical templates, 3D printed implants for veterinary use. We have not entered human implants and human market as for now, because we have focused mostly on veterinary, which is far more interesting for us, saving different species and providing the services for really vast amount of dogs, cats, and other species which need such help. We have anatomical models which are completely useless by means of surgeries, but the idea is to educate the customer of veterinary surgeons. It's quite often found that people don't understand what is the problem in their pets, and veterinarians don't want to draw or use pictures every single time. So we are providing models of different parts of the bodies, different parts of different animals. And all of them are really high quality because they are made either on a scanning of cadavers or on a basis of CT scanning. So not only models or help during the surgeries, but also this business side of veterinary market, so educating the customer. We are also designing some devices related to the needs of the doctors. So if they need some, as here, anesthesia-free flexible anesthesia tube holder, which will be resistant to UV sterilization in the surgery room, one-time single project, just finding the problem which have the surgeon and solving it, then incorporating it into our offer. But also we have this sport department, so R&D for medical, but also sport department. So custom-made protectors, 3D printed or manufactured using thermoplastic materials, made on a basis of 3D scanning or CT scanning. As you can see here, for example, a custom-made sport mask for the player of our local football club. And the idea is to create such a model as fast as possible, because using 3D scanning and 3D printing, we can create it within 2 working days. Normally, when you have to cast the face, it's rather 1 or 2 weeks instead of days. Moreover, we are going into cooperations with club, like scanning players before the injuries. So we are building our own database of players, and if some of them will have injury in an area of face or skull, we can create a protector within 1 day, even if the player will not be here. So prevention instead of firefighting. Our protector was playing in Euro 2020 qualification because one of the players is a representative of Estonia. But the idea is to talk about custom-made mass-scale manufacturing. I have 2 examples which will describe what I mean exactly. One of them is a project of our customers who had only the brand and idea, so we had to create all of the technology behind the product. The second one is, I would say, not so obvious, but it's pure WEBINAIRE project. The company quite known in Poland is for now base prosthetics, so 3D printed hand prostheses made in SLS technology, so selective laser sintering. Design product. Actually, the idea is not to be completely functional, to have movable fingers. The idea is to have some type of Apple in a world of prosthetics. And that's why Glaze ordered us to create, for example, hand with built-in Bluetooth speaker. So for us, it's not a problem. They had idea, we could create it. But at the beginning, the first model, which was prepared by a student of art school, was fit only for the one patient. So they had only one size and would like to go into the market. So there was a one size and design requirement for developing such project. So the main task was to provide a solution which will answer to the needs of at least 80% of population, because every hand is different. So the size of every hand is completely different. From the other side, we cannot create millions of combinations because it was crucial to have also this business side, so optimization of costs and also optimization of manufacturing process. So on the basis of anthropometric data analysis, we have assessed that it will be needed to have 7 sizes of palm part, 7 sizes of wrist, 5 elbow sizes, and the range of forearm length and circumference, as you can see. And we have a lot of combination. We had a lot of combination at that time, because on the basis of those sizes, we have got more than 2 million of combinations of one hand. But we have right and left hand, so it was almost 5 million of sizes. And Glaze had 2 designs. So at the end, we created the idea to have a system which will allow to provide 9.5 million of combinations of hand size to provide perfect fit. But then we have realized that it is quite not possible to have palm of extra small and elbow of extra large. So we have created sizes combinations, and in the end, we have finished with 30,000 of combinations of one hand, and the design is created within 1 hour. So we have set up the whole technology, the whole system, that from the order to creating files for printing, it is less than 1 hour. Then the design is printed, assembled, painted, and etc. I would say, because going from one size to 30,000 of combinations of one hand, so in total 120,000 combinations, was also connected with other technological tasks. So how to print it, how to finish it, how to assemble it, providing quality control, and the way of the delivery. So as a subcontractor, which is somewhere in the background of Glaze Prosthetics project, we have created the system from one size to fully functioning manufacturing process. And now Glaze is maintaining it by themselves. So our task is to create any other projects related to, for example, Bluetooth speaker, etc. We could also create prosthetics for below elbow amputation, which is made on a 3D scanning, and the system is also prepared in that way that you don't need to design it for a couple of days. Full hand is designed below 3 or 4 hours, even if it's needed to provide to design it on the basis of 3D scanning. A bit different approach to custom-made mass-scale manufacturing, which is really close to our core business as for now. It's our product, so it has WEBINAIRE brand, and at first it's not so obvious area of medical market, and the idea of the product was, I would say, pretty silly, because it seems to be really easy to manufacture, but it's not. And I'm talking about wheelchairs for small animals. As we are working with veterinary market, we have found that there's a need of some different product that it's now available, because all of the wheelchairs available for animals in the market were either big or heavy or not so tailored for the small animal. In the right, you can see the lightest wheelchair which was available in the market. its weight is 1.5 kilo, whereas our patient was 3.2 kilos. So it was 50% of its mass. So then we have decided to create some product which will fit into this custom-made mass-scale manufacturing. So we have created our own wheelchair, which is partially 3D printed, involving carbon tubes, really fast wheels, really fast bearings. So from 1.5 kilos, we have go down to 500 grams. So from 50% of body mass, we have now 15% of body mass. And the wheelchair is completely customizable by the end user, because there is a huge potential in regulation, huge potential in fitting it into the patient, even if it's made on a measuring protocol, because we are using 14 parameters of a small dog, and the wheelchair is made almost automatically. We have created the files and the system that instead of designing it for example, 2 weeks, we can start production within 2 hours. Then it has to be 3D printed and assembled. So we can create tens of wheelchairs completely custom made within 1 week, instead of designing it for 2 or 3 weeks as it was in the past. So as I said, we have file parameterization and process automatization, highest level of comfort because of custom-made 3D printed shell on a basis of measuring protocol. Ultra-light design, less than 10% of patient body mass in the end, because we have created more and more lightweight designs and wheels in our product. And the idea was to provide also easiness of wheelchair operation, not for a pet, but for pet owner, because it was a problem with other products available on the market. So, custom-made mass manufacturing with proper approach to using 3D printing and possibilities of file parameterization, we can create a system for the product, either our product or our customer product, which will allow to fully parameterization within some range of anthropometry, and it will be available for manufacturing with, for example, 1 or 2 hours. In the glaze case, we can fit 30 prostheses in 1 printer batch. So for 2 days, we can create 30 designs of completely custom-made project, which is laser sintered. So it was not possible without 3D printing. It was not possible without huge file parameterization and optimization. And from the one hand, rehabilitation for people. From the other hand, rehabilitation for animals. Principles are completely the same. The idea was to create completely custom-made product, but it will behave like mass production. In case of dog wheelchairs, we have more than 1 million combination of sizes on the basis of measuring protocol, and we are focusing only on pets below 10 kilos. So yeah, that's the idea of custom-made mass production. It could be used also in a sport, because we have a couple of projects related to sport protectors, but it's a bit confidential as for now. And as you can see, our patient is really pleased with the solution we have prepared for it. And
so, excuse me for interrupting you. Now we are at the end of the webinar. Do you have, is this the last slide of you?
Yes, that's the last slide. Here you can find me, call me, or send me some email.
Okay. So then I would like to thank you for this really impressive talk and how you use 3D printing in your company. And now I don't know if there are any questions from your side. I can't see it in my chat function. Maybe Emily, you do you have any question on this? No? So
No,
then okay, then there are no questions, so you have the chance to send us your question and we forward it to our speakers. And I hope you have enjoyed this webinar and get valuable information on this technology. And hopefully we will see you in the next webinar or in our event in Tübingen. So thank you and goodbye.
Thanks a lot. Bye.
Thanks. Goodbye. Have a nice day. Bye.
Thank